Hey students, it's Mr. Lucido. Just wanted to create this quick video recording the lesson from earlier today. Remember that this lesson had two objectives. The first was to consider the different variables and control groups that are in experimental design. And number two was I applied it to a specific experimental context that's related to what we've been studying in Unit 3. Okay, so we're going to start with just refreshing our memory of the experimental context. So if we were in person, we would be doing this lab right about now. And this lab was all about testing enzyme function. And so the question that's going to be addressed by our experimental context here is, how does temperature impact catalase function? Now we need to know that catalase is an enzyme. It's a protein that catalyzes a reaction, and specifically the reaction that this enzyme catalyzes is the breakdown of hydrogen peroxide into water and oxygen. Now in class I made the connection that, remember that the peroxisome, right, is responsible for generating hydrogen peroxide as a byproduct of some of the reactions that the enzymes inside of the peroxisome are involved in. And hydrogen peroxide is actually toxic to the cell. And so also in the peroxisome, you'll find catalase, which helps to break that down before it becomes toxic to the cell. Okay, so with this in mind, we're trying to test in this experiment, how does temperature impact this specific enzyme's function? Okay, now what we can start with are two variables that are familiar, independent and dependent variables. So if we think about this question, we can identify our independent variable as being the thing that will have to be changed intentionally throughout the experiment in order to test our question. And so based on our question, temperature is going to be that variable that we're intentionally changing in order to answer our question. Now, what are we going to be measuring? Well, that's going to be our dependent variable, and based on the question, it is catalase function, which can be also measured in terms of the rate of the reaction that it's catalyzing, right? That's going to tell us about how well or not well catalase is functioning. Okay, so with those two variables in mind, I want to tell you a little bit more about this experimental setup. Now, this lab, which I think is pretty fun, is called the Floating Disk Lab. And the way we set it up actually is we can extract catalase, and that's what we see right here, this kind of cloudy solution. This is catalase that's actually been extracted from yeast. And what we can do is we can fill a cup with some hydrogen peroxide, and we're going to take a little paper disc. It's often used, we use filter paper, and we'll take that little disc right here, and this is soaked in catalase. So we would dip it into that solution, right? That's over here, catalase solution. And then we would drop it into the hydrogen peroxide, which immediately is going to start this reaction as soon as it touches the hydrogen peroxide. Now, the way that this is useful is that it can actually help us to measure the rate of catalase function. If we take a look down here, this is how we do that. You see, when you drop this disc into the liquid, it's going to sink to the bottom. And as soon as it hits the bottom, what we can do is we can start that timer, and we're going to determine how quickly does that paper disc float back to the surface. Now, why does that tell us something about catalase function? Well, if we think about what catalase is doing, it's breaking down hydrogen peroxide into water and oxygen. So actually what you would see if you did this in person is that you would see lots of bubbles start forming all around, and they're actually oxygen gas bubbles. And the faster that catalase is functioning, or that reaction is occurring, the more oxygen is going to be produced, and therefore the more bubbles. And those bubbles are what are responsible for pushing that disk back to the top of the surface. And so this can be used as an assay, a tool for measuring catalase function, or in other words, the rate of the reaction that catalase is helping along. Okay, so we use the floating disk lab to help us to measure the speed at which this reaction is occurring, with the faster time being associated with a faster rate of reaction, and a slower float time being associated with a slower rate of reaction. Okay, so with that in mind, we have a means of measuring our dependent variable. Okay, now the other type of variable that's present in an experiment are constant variables. Now, constant variables, as their name implies, are variables that are kept the same throughout the entirety of the experiment. And so there are multiple constant variables that we need to account for in order to really effectively test the relationship between our independent and dependent variables. So just to name two, one thing that should be kept constant is the concentration of solute, substrate. Okay, now just to review, right, what's the substrate of this reaction? Well, it's going to be hydrogen peroxide. That's what catalase is breaking down. And if we were to look at the molecular level, hydrogen peroxide actually binds to the active site of catalase. Okay, the second thing that we're going to want to keep constant is the concentration of enzyme. All right, these two things, in this case, catalase is our enzyme, these two things should be kept constant in order to focus on how does temperature affect the rate of this reaction. Right, because we know that changing the substrate and changing the enzyme concentration can affect the rate of the reaction that's occurring. Okay, now you can also think of a few other constant variables like pH or salinity, or a couple other things that should be kept constant, but these are the two that I mentioned this morning. Okay, so now that we have our variables, we can think about our experimental and our control groups. Okay, now our experimental groups right over here, these are going to be the main element of this experiment. Right, the main element of this experiment is we're testing how do different temperatures impact catalase function. And so in class, I mentioned that we could set up 3 experimental groups where we have 3 different temperature conditions. Right, and again, the purpose is we're testing the effect of changing temperature on enzyme function, right? This is kind of the main part of the experiment. And so at 20 degrees Celsius, at 40 degrees Celsius, and 60 degrees Celsius, we would run our experiment, right? Like we see right here. And we would see, we want to measure the rate of reaction for all of these situations, right? And again, we're measuring that by looking at how fast the disk floats to the top of the surface at 20, 40, and 60 degrees Celsius. Okay, now you could obviously add lots of other temperatures to test at, but hopefully you see that the purpose of the experimental group is to really get at the relationship between temperature and the rate of the reaction. Okay, so then what are these control groups? So control groups can be thought of in 2 separate categories. So every good experiment has positive control groups and negative control groups. And in class, I summarized first kind of the purpose of these two, what are they trying to accomplish, and then I applied it to this context. So let's start with a positive control group. So a positive control group is attempting to verify your experimental procedure, meaning that you're going to carry out this experiment in a way where you know that if your experimental procedure is working, in this case, the floating disk assay, if it's working, then you should get the result you expect. If you don't, then that means something has gone wrong in your experimental setup. So what would that look like? So for this experiment, a common positive control would be, we know that catalase functions, this type of catalase, it functions at body temperature. This is the same catalase, I know I mentioned it's from yeast, it's the same catalase that's found in humans. It operates at 37 degrees Celsius. We know that because that's the temperature that's inside of our bodies and also that yeast commonly live at. So about 37 degrees Celsius, we know that this enzyme should work. So at the very least, we should get the disk floating to the top of the surface, right? So we're going to run the experiment at 37 degrees Celsius, and we expect that the disk should float. If it doesn't float, that means something is going on, meaning that this is not an accurate way to measure catalase function if we can't get it to work at 37 degrees Celsius. All right, so that's the purpose of a positive control, is you should have a positive result, meaning that you run the experiment with an expected result, and if you don't get that, then something is wrong. Okay, a negative control. Typically, a negative control is intended to identify any confounding variables, meaning anything else besides what you're testing for that might be impacting your results. That might be giving the appearance of a result, but is actually caused by something that you haven't accounted for. So the way that we set up a negative control is we are going to intentionally leave something out to make sure that we're not getting any false results. So what that would look like in this experiment is for every one of these temperatures, we're going to have a negative control group where we're going to run the experiment with no hydrogen peroxide or no catalase. All right, so let me elaborate what I mean by that. So at 20 degrees Celsius, you'll run your normal experimental group. You'll measure the rate of reaction. And then your negative control is going to be at 20 degrees Celsius. Let's say we choose this first option. At 20 degrees Celsius, I'm going to run a negative control where instead of using hydrogen peroxide in the cup right here, I'm going to use water, meaning there's no substrate. I'm still going to dip my paper disc into the catalase. I'm going to drop it into the cup of water. And if everything is accounted for, that disc should just float to the bottom. It should not float back up. I'm sorry, it should sink to the bottom. It should not float back up. Right? If it does, that means something else is going on that's causing the disc to float up, other than what I am intending, which is to measure catalase function. Right? So no hydrogen peroxide, or let's say instead of catalase, right, you dipped the paper disc into water and then dropped that disc into hydrogen peroxide. Still, you should see that disc float. You should see that disc sink to the bottom. It should not float back up. Right? If it does, there's something else that you haven't accounted for. All right. So a negative control. The reason it's called a negative control group is because
you are expecting a negative result, meaning nothing should happen. If something happens, then there's something else you haven't accounted for. Okay. Both of these in an experiment, a positive and a negative control group, in addition to your experimental groups, are going to solidify your evidence by removing any alternative explanations for the results that you get. All right, because if you think about the results that you're going to get, right, you're going to have the relationship between temperature and the rate of reaction, right? You're hoping to find at what temperatures, we know that it functions at 37 degrees Celsius, but what other temperatures does it function at, and where does it begin to denature or not function anymore? Okay, and so we want to make sure that we've accounted for everything so that we can assume that our results are strong evidence. Okay, so I hope that was helpful. I know that these can be a little bit tricky, positive and negative control groups, to differentiate, and we'll get some more examples throughout this unit. But please reach out if you have any questions. I hope you have a good rest of your afternoon.